Today I'll be discussing the unit 11 in biophysical chemistry. So in this unit we'll be that unit 11 mm, mainly deals with the basic concepts of enzyme kinetics. So what is enzymes? Enzymes ಅಂದ್ರೆ ಏನು? It is an protein that helps us to speed up metabolism or the chemical reaction in our body. So enzymes are nothing but the proteins. What are proteins? Proteins are building blocks of amino acids. Amino acids you should be knowing the 21 amino acids present. So proteins are building blocks of amino acids, and also enzyme can be also known as proteins here. Enzymes are nothing but proteins that help us to speed up metabolism. So here we are going to study about the proteins and their reactions. Enzyme kinetics. Kinetics ಅಂದ್ರೆ ಏನು? In previously we have studied about the chemical kinetics. Chemical kinetics ಅಂದ್ರೆ at the rate at which the reaction is taking place. So that is what with respect to chemical kinetics. Ille we are going to study about the enzyme kinetics. Enzyme kinetics is nothing but the studying of biochemical and biophysiological process. It is necessary to measure the rate at which the reaction is taking place. So biochemical and biophysical process we are going to study. Enzyme kinetics is nothing but the protein and ligand interaction. Protein and ligand interactions we are going to study at what rate the reaction is taking place and whether it is having binding affinity. So this part we are going to discuss in this unit 11. So what are the main topics we are going to discuss here, Andre? The basic concepts of enzyme kinetics. What is enzyme kinetics anta? It is the study of rate of enzyme catalyzed chemical reaction. So it's nothing but the protein and ligand interaction we are going to study the rate at which the reaction is taking place. Enzyme kinetics andre. And catalytic antibody and RNA enzyme that we are going to study, and mechanism maintain kinetics. There one derivation is there, and computation and inhibitors. What are computations and inhibitors? And Moon Wyman change X mechanism and substrate as inhibitors. Substrates are nothing but ligands. So effect of chromium, iron, zinc, zeolite, etc. by adding of those catalysts, how the reaction, whether the rate of reaction is increasing or decreasing, will be studying. And also in the presence of if the reaction is taking place under the UV light, what are the changes? If the rate of reaction increases or decreases, that we are going to study in this. This being biophysical mainly with the physical physical process or biochemical or physiological process. It is necessary to measure the rate at which the given reaction or process proceed to completion. So the rate at which the reaction will be taking place, that we are going to study in this unit. So these are the introductions. Go through this. Here, enzyme kinetic is the study of rate of enzyme catalyst chemical reaction. So, what are enzymes? Enzymes are nothing but proteins. Proteins that helps us to speed up metabolism. Proteins which help us to speed up metabolisms are known as enzymes. Enzyme kinetics is the study of rate of enzyme catalyze chemical reaction. So it is mainly enzyme kinetics is nothing but the protein ligand interaction that we are going to study the rate at which the reaction is taking place. So an enzyme is typically a protein molecule. So what I said before earlier, the enzyme is typically a protein molecule that promotes a reaction to another molecule. Its substrate. It's nothing but protein and ligand or protein substrate. Means ligands can be also called as substrates. Here, don't get confused. Proteins andre enzymes. So enzymes andre proteins. So substrate andre ligands. Don't get confused. For example, here they have taken E plus S, which is reversible reaction forming of ES. Enzyme plus substrate. This one. Enzyme plus substrate gives enzyme substrate. Further forming enzyme substrate intermediate forming East E S star, that is the intermediate formation of intermediate, further forming the product enzyme plus product, and further separates to give enzyme separate and the product will be separate. So these are the steps what the reaction is taking place. Enzyme and substrate andre it's nothing but the protein structure plus a drug molecule or a ligand, a chemical composite, whatever the taken, a structure or a reactant you are taken as a substrate. So enzyme plus substrate andre protein plus an ligand. So forming enzyme substrate next in the next step ES star, which is known as enzyme substrate as an intermediate forming the product enzyme product, and further substitute to give enzyme product. So the bind it binds to active site of the enzymes to produce enzyme substrate complex, that is the ES complex. Further formation of enzyme plus product. So here some mechanism maintain concepts have been applied, and that is nothing but substrate forming enzyme substrate and further forming uh, product. So it's nothing but the protein and then ligand interacting, further forming an protein substrate and ligand is separated. So this Michaelis Menten kinetics is a mathematical model that describes the rate of reaction. Enzyme reaction andre ide enzyme plus ligand forming the enzyme plus product. So this is the rate of enzymatic reaction, and it was proposed by Leonard McLeans and Maud Maintain in 1930. So here the substrate bind to form complex. It's nothing but the substrate and enzyme bind to form complex, which undergo chemical reaction to form the product. So was the reaction. The substrate bind to form complex with the protein, and which further it undergo chemical reaction to form the product. So this was the theory explained by Michaelis Menten. So basic concepts of enzyme kinetics. Kinetics andre what are the basic concepts they have explained here. In this, enzyme kinetics is a study of chemical reaction. So chemical kinetics andre it's a chemical reaction. So enzyme kinetics, it's a biological reaction that are catalyzed by enzymes. Basic concepts of enzyme kinetics includes Maclaurin equation and the concept of saturation, which occurs with enzyme operating as maximum capacity, and the rate of reaction is no longer independent on the substrate concentration. What this theory explains andre the rate of reaction no longer depends on the substrate concentration. Substrate what we are taken here in the reaction, this is the substrate. E concentration ಮೇಲೆ depend ಆಗಲ್ಲ after certain time. When the concentration of this substrate is increased, the rate of reaction also increases until certain time. Later on, it won't affect. That was decided by Michaelis Menten. So that theory was the proposed here. In detail, we'll explain in the next uh, topic. And the concept of enzyme inhibitors, which occurs when molecule bind to enzyme and reduces its activity. So the molecules which bind with the enzymes and the molecules which bind with the proteins and reduces its activity. So this is usually quantified using dissociation constant, which is substrate concentration at which enzyme is after saturated. So this concept will be explained in detail in the next topic. And enzymes are complex nitrogenous substances, are proteins, but may be associated with non substances. For example, here they have taken enzyme catalyze most of the biochemical process. Enzyme catalyzes most of the biochemical process, such as digestion, biosynthesis, etc. in animal and human being. So enzyme kinetics andre it is mainly for biochemical process. Chemical kinetics andre general reactions, chemical reactions which is taking where which we are going to conduct an experiment. So those are known as chemical kinetics. Enzyme catalysis or enzyme kinetics andre which is taking which is undergoing inside the body or in an animal. So like digestion, biosynthesis, and enzyme catalysis most of the biochemical it is mainly of biochemical process. And for example, sucrose gives glucose plus fructose. Uh, Here it cannot be breaking of maltose. Means this is the process which is taking place inside the given small example for glucose. And one more enzyme reactions are much sensitive to catalytic poisoning such as hydrogen cyanide, hydrogen sulfide, and carbon disulfide, etc. So the reaction may reduce or it may catalytic, it may be poisoning. So it is uh, hydrogen cyanide, hydrogen sulfide, carbon disulfide. These are very poisonous to living beings. Ala. So these are sensitive and catalytic poisoning, such as hydrogen cyanide, hydrogen sulfide, and carbon disulfide. So these chemicals, where it affects the enzyme reaction and also in the poisoning of the cells. And activity of this enzyme depends upon the non-protein substances. Also, with respect to coenzymes, uh, nucleic acids, RDNA and RNA strands, coenzymes andre. So for each enzymes, there is only one coenzymes. Enzymes loses their activity in the presence of electrolyte when exposed to UV radiation. So generally, if the reaction is taking place under the UV, it you know it loses their activity. So means the rate of reaction will decrease when it is exposed under UV radiation. So this point is very much important. So catalytic antibodies and RNA enzyme ribosomes. So the scientists use genetic engineering technique to create antibodies. Antibodies were generally created by genetic engineering. So and catalyzers specific of chemical reactions. So antibodies were generally created by chemical reaction and also in the genetic engineering. So catalytic antibodies have wide range of potential applications in various industries such as biotechnology, pharmaceutical companies, and medical research. So there can be antibodies andre ಏನು an antibody is a protein component of the immune system that circulates inside the blood. So it is an protein component. Some of the antibodies it can be injected. So this can be used as a drug to target specific enzymes also. Some of the proteins na treat ಮಾಡೋದಕ್ಕೆ it can be used as a drug. Antibodies can be used as a drug to target specific enzymes. Enzymes are nothing but the protein which is in the disease process. So as enzymes to catalyze the specific reaction in industrial process, and also diagnostic tool to detect the presence of specific molecules in an sample. So inside the protein structure, there will be an some repaired or damaged area where by utilizing the drug, by the usage of drug, it can be repaired. The enzymes in the uh, body can be repaired or by induced. So, what is antibodies, Andre? It is the antibody, a protein component of the immune system that circulates inside the body. So, next, what is ribosomes, Andre? Ribosomes are RNA enzymes, which are molecules that are capable of catalyzing chemical reactions. Similar to proteins. And they are composed of ribonucleotides and the building blocks of RNA. So ribosomes are nothing but the building blocks of RNA, which allows to have enzymatic activity, means which is having the same protein activities even the ribosomes are having. So coming to the main topic, this is very much important. Make maintain kinetics. So the equation describes the relationship between the rate of enzymatic reaction and concentration of the substrate and the enzyme. So this equation states that the rate of enzymatic reaction is directly proportional to the concentration of the substrate and the product concentration of the enzyme and the binding of enzyme substrate. So this definition I will explain here. It will be in the plot. So here in the x-axis we have taken a substrate. Substrate andre it's nothing but the molecule which is reacting with the protein or also reacting with the enzyme. So as the concentration of the substrate is increased, ಈಗ ಯಾವ್ದಾರ ಒಂದು ಮಾಲಿಕ್ಯೂಲ್ ರಿಯಾಕ್ಷನ್ಗೆ ತಗೊಂಡಿರೋದನ್ನ ಅದ್ರ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಇನ್ಕ್ರೀಸ್ ಮಾಡ್ತಾ ಹೋದ್ರೆ the rate of reaction also will increase. Here the rate of reaction, where the rate of reaction which is taken in the y-axis, as the rate of reaction also increases when the concentration of the substrate is increased. ಇಲ್ಲಿ 1 2 3 4 ml ಆ ಥರ increase ಆಗ್ತಾ ಹೋದ್ರೆ substrate concentration, the rate of reaction also will increase up to certain time. Later on it become stable. So further, as the substrate concentration increased also, there will be no change in rate of reaction. So that concept was described by Michaelis-Menten kinetics. So here this is Vmax is the maintain velocity. So Km is the Michaelis constant. To how you are going to calculate the constant Km andre, so plotting against the graph, taking Vmax ಎಷ್ಟು ಬಂದಿರುತ್ತೆ ಇಲ್ಲಿ ಅಂದ್ರೆ Vmax is nothing but the rate of reaction which will become maximum, means after that it will be constant. ಎಷ್ಟೇ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಸಬ್ಸ್ಟ್ರೇಟ್ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಇನ್ಕ್ರೀಸ್ ಮಾಡ್ತಾ ಹೋದ್ರು the Vmax will become constant. So that Vmax will, as the concentration of the substrate is increased, the Vmax, the rate of reaction will become stable and it will be constant. So that Vmax, half of that Vmax is known as Michaelis constant. So for example 4 ಅಂತ ಬಂದಿದ್ರೆ rate of reaction, so half of that ಅಂದ್ರೆ it will be ಏನದು 2. For the 2 you are going to plot the slope for that graph, and you are going to calculate the km ಎಷ್ಟು ಬಂದಿದೆ you are going to, that is the Michaelis constant. So here you are taking the example for the reaction. So that is what explained from the Michaelis Menten kinetics. So enzyme plus substrate gives enzyme substrate. Enzyme substrate andre it's nothing but the protein plus molecule or a reactant. Protein plus substrate gives enzyme substrate, further forming the product plus enzyme. So that is the general equation, general reaction taking place in this concept. So K1 and K minus 1 are nothing but rate of reaction. K1, K2 are rate of reaction. Next, how you are going to calculate the rate? First step rate of reaction of K1 and K minus 1. That product minus reactant you are going to calculate. ಈಗ same ಈಗ rate you are going to calculate for the both the steps. For the second step, rate K2 is equal to K2 into enzyme substrate. So enzyme substrate are nothing but the product form. That is the equation 2. Applying steady state concept, d by dt is equal to K1 into enzyme substrate minus K minus 1 into enzyme substrate plus K2 into enzyme substrate. That is the equation 3, that you are going to calculate the rate of formation of enzyme substrate or rate of reaction generally. So next step you are going to take it on left hand side and further substituting for enzyme substrate the K1, K1, K2, K3 are the rate constantly. So substituting the equation 4, the equation 4 in equation 2. So in this equation we are going to substitute this, further substitution ಮಾಡಿದೀವಲ್ಲ, so that one you are going to here, that we are going to substitute K2 into K. This one you are going to insert, that is the equation 5. Or the equation can be written in the rate. So rate is equal to Km into enzyme substrate. Km is nothing but another Michaelis constant. So Michaelis constant, what you are going to calculate? Km is equal to E and enzyme the E ಮತ್ತೆ S to value ಗೊತ್ತಲ್ವಾ? This is the ES. So that one you are going to insert in this ES in the equation 6. Then you are going to get the Km is equal to K2 into K1 by K2 plus K minus 1. So that is for enzyme substrate value that we are going to insert, and we are going to calculate the rate of volume. So rate of that total reaction K2 into E naught S naught by Km plus S naught. So that is the equation 7. So V is the velocity of the reaction, K2 is the maximum rate achieved by the system, and concentration of the substrate. Km is the Michaelis constant, and S naught is the concentration of that substrate. So while varying the concentration, here you have seen concentration of the substrate has been varied ala. So we have got a general equation, that is equation 7. So that we are going to study in different cases. In the case 1 only, when the substrate S naught is low, concentration of the substrate S naught is low on the glow, the rate of reaction is increasing. So that we can see in the plot. As the concentration of the substrate is increased, the rate of reaction also increases. So here the rate is independent of substrate concentration. And in also in the second case, We are going to see at high S naught, at high S naught andre the concentration of the substrate is very high. So in the second case, how the plot is shown here. ಇದು high S naught ಅಲ್ಲಿ ಬಂದಿರೋ plot. So in this plot, the concentration of the substrate is maximum and the concentration of that molecule is maximum andre it will reaches the rate of reaction reaches to a certain point and it will be constant. So the it will be constant when the concentration of the substrate is maximum andre it will be constant and it will be independent of the reaction andre after certain time while increasing it will be in the relation with the rate of reaction after certain time ಅಂದ್ರೆ certain time ಆದ್ಮೇಲೆ concentration ಇನ್ನು extra add ಮಾಡ್ತಾ ಹೋದಾಗ the rate of reaction will be constant. So here rate is dependent on the substrate concentration. The significance of equation ಅಂತ ಇದೆ. So Maclaurin-Mentini equation is can be written also in this pattern. 1 by V naught is equal to 1 by V max plus Km by V max into 1 by S naught. V max is the rate of I volume at I concentration or V max ಅಂತ same same plot they are given in the 1 by V naught, that is the rate, the concentration of substrate 1 by Sn. This is the concentration of the substrate. So same equation they are written in. So if you go through this, for example, they have taken enzyme plus substrate, giving the enzyme substrate further forming enzyme substrate intermediate, find formation of enzyme plus product. So for this, the rate is given by same. They are the one step, the intermediate of enzyme substrate has been noted here. So it can be written as V is equal to V is nothing but rate at high substrate. So the rate of reaction when the concentration of substrate is very high andaga. So that can be written as V is equal to K1 into K1 dash. K1 in K andre ಏನು? These are nothing but the rate of reaction of first step to second step and second step to third step. K1, K1 dash, K2. Those are nothing but the rate of reaction. E is for enzyme concentration, E naught, and S naught is the concentration of the substrate. ಇದು. So next, we are going to study with effect of pH. pH ವೇರಿ ಮಾಡಿದಾಗ ಈಗ ಸಬ್ಸ್ಟ್ರೇಟ್ ಈಗ ಒಂದು ಮಾಲಿಕ್ಯೂಲ್ ತಗೊಂಡಿದೀವಿ ಸಬ್ಸ್ಟ್ರೇಟ್ ದಟ್ ಕ್ಯಾನ್ ಬಿ ಡಿನೋಟೆಡ್ ಆಸ್ ಸಬ್ಸ್ಟ್ರೇಟ್ ಅದ್ರದ್ದು ಕಾನ್ಸಂಟ್ರೇಷನ್ ವೇರಿ ಮಾಡಿ ವಿ ಆರ್ ಸ್ಟಡಿ ದ ಪ್ಲಾಟ್ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಚೇಂಜ್ ಮಾಡ್ತಾ ಇನ್ಕ್ರೀಸ್ ಮಾಡ್ತಾ ಹೋದಾಗ ದ ರೇಟ್ ಆಫ್ ರಿಯಾಕ್ಷನ್ ಆಲ್ಸೋ ಇನ್ಕ್ರೀಸಸ್ ಆಫ್ಟರ್ certain time it will become stable. So same thing while varying the pH of that substrate, pH concentration change ಮಾಡಿದೀವಿ. Now we are going to change the pH of that substrate. ಏನಾಗುತ್ತೆ when the pH value of that means the pH value of the substrate is increased, it reaches maximum catalytic activity. So you can see maximum. So the rate means the rate of reaction will increase up to certain and decreases after the increase in further pH. So generally optimum pH andre, so this pH will be around 7 plus or minus. So the substrate to pH it should be around 6 to 8 for the reaction taking place. That is the effects of pH. So an enzyme can exist in 3 different states of ionization, EH plus 2, EH, and depending on the pH. also E plus corresponding with substrate. EH NCHS is more active than EH plus 2, and these are in the different state. So this EH2, EH and E minus. So this is also formation of the product if they are in different state. And also with respect to temperature, the substrate to temperature ವೇರಿ ಮಾಡಿದಾಗ ವಾಟ್ ಇಟ್ ವಿಲ್ ಹ್ಯಾಪನ್ ಅಂದ್ರೆ ಕಾನ್ಸಂಟ್ರೇಷನ್ ವೇರಿ ಮಾಡಿದೀವಿ, ಪಿ ಎಚ್ ವೇರಿ ಮಾಡಿದೀವಿ. ನಾವು ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು vary with respect to temperature. As the rate of enzyme catalytic reaction increases with temperature, reaches maximum, then decreases. So when the temperature is increased, the rate of reaction also increases. Later, then decreases after certain. So it mainly depends on the temperature. E temperature ಇದೆಯಲ್ಲ. So what will be the maximum temperature for this reaction to take place andre until 35 degree Celsius. So why andre after that while biochemical process is taking place andre inside the body andaga the cells will be live up to 35 degree Celsius. Later on when the it is increased, the cells will be died. So the general enzymes are stable at lower temperatures. The proteins are very stable at lower temperatures. So cells ugly, proteins ugly, whatever it may be stored in the cold storage. It will be around minus 15 degrees Celsius ಆ ತರದ್ರಲ್ಲಿ ಸ್ಟೋರ್ ಮಾಡಿರ್ತೀವಿ. If you want to keep it for a longer time ಅಂದಾಗ when the temperature increase it get died. So so generally enzymes are stable at low temperature and catalyzes the reaction. So the temperature greater than 35 degree, the kinetic energy of enzymes get exceeds to energy barrier for breaking of weak hydrogen bond and hydrophobic bonds. And the competition and inhibition. What is competition and inhibition? Andre refers to interaction between different molecules or enzymes in a biological system. So inhibit andre which inhibits competition which observes anta illi. So competition occurs when two or more molecules or enzyme complete for the substrate or a binding site. So when two drugs that bind together uh, with the receptors. Here you should not get confused. Drugs andre receptors andre should be receptors are nothing but the proteins. Proteins are nothing but the enzymes. It can be called in all the three. Enzymes ಅನ್ಬೋದು, proteins ಅನ್ಬೋದು, ಇಲ್ಲ receptors ಅಂತ ಅನ್ಬೋದು. And coming to another part, substrate andre substrate it can be molecule also it can be called. Also it can be called as drug molecules. So it should be knowing difference between the receptors and the substrate. Receptors, so proteins, so enzymes, all are same. And coming to another part, drugs, molecules, are substrates are same. Should be knowing these differences. So true drugs that bind to same receptors means two drugs that bind to same receptors may complete for the same binding site. So two drugs, two drug molecules, it is binding to a same receptor andre it is nothing but the protein. When the drug molecule is binding with the protein, it completely forms same binding site, means it get binded. So leading to decrease in the effectiveness one or both the drug. Also, it occurs when a molecules or enzyme interferes when the activity of the another molecule enzymes. This can occur through various mechanisms as substrate completing by and allotropic regulations. Allosteric regulations are mechanism by which molecule bind to specific site. For example, this will be a protein structure. Here there will be a binding site. So here one more binding site will be there. So when you take a insert a drug to that living organism, it's get binded in one drug site illa another binding site ಅಲ್ಲಿ protein structure ಅಲ್ಲಿ gets binded. So this is the actual formation of that protein binding with the drug molecule. So allosteric regulation is a mechanism by which molecule binds to specific site on enzymes. Enzymes are nothing but the proteins. Is called allosteric site, and the allosteric regulations is nothing but the molecule bind to specific site. The molecules bind to specific sites of enzyme is called allosteric sites. So next, here one more mechanism was discussed in this moon Wyman change mechanism. So in this model, proposed to explain mechanism of allosteric regulations in protein. So in the protein structure, the allosteric regulations, allosteric ಅಂದ್ರೆ specific binding site ಗೆ ಹೋಗಿ bind ಆಗಬೇಕು. Ligand molecule or a drug molecule or a substrate. See what are the terms we are going to use ಅನ್ನೋದಕ್ಕೆ. Substrates ಅಂದ್ರು ಒಂದೇ, ligands ಅಂದ್ರು ಒಂದೇ, drug molecule ಅಂದ್ರು ಒಂದೇನೆ. Should be knowing this difference. And coming to another part, protein andro one day, enzymes andro one day, and one more word which you are going to use, Andre, that is allosteric regulations in the protein. So we should be binding to a specific active site. So the conformation of these states are referred to as active or inactive state. So here uh, the conformation of states, it can be denoted in the two forms. ಈಗ ಪ್ರೋಟೀನ್ ಸ್ಟ್ರಕ್ಚರ್ ಇದೆ. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಯು ಆರ್ ಟೇಕಿಂಗ್ ಸಮ್ ಪ್ರೋಟೀನ್ ಸ್ಟ್ರಕ್ಚರ್. Here there is a two active sites. So when one drug molecule is interacting to this, see, these two are the active sites, and this is the drug molecule. It should interact with any one binding site of that protein molecule. Other one binding site ಗೆ ಹೋಗಿ bind ಆಗಬೇಕು. So bind ಆದಾಗ whether it is in active state or inactive state, it can be, if here it gets binded or if it here it gets binded andre, one will be active site, another will be inactive state. So whatever that, we should find the difference. So this mechanism was introduced here, moon Wyman change mechanism. So in this model, the allosteric proteins are made up of multiple subunits, each of which can exist in either active or inactive state. So the binding of ligands or drug molecules to the protein can change the equilibrium between the active and inactive state to subunits, leading to change in overall activity of the protein. So the equilibrium can be changed when one drug molecule is binded to active one binding site. So it can be whether it can form the protein may become active or it may be inactive depending on the sites where it is the drug molecule is getting linked or binded. So that was the theory described by this moon Wyman changes. So here the allosteric proteins is an oligomer, the promoters that are symmetrically related where shall we sake of algebraic sum, and it is in the form of hemoglobin anta. So here the promoters can exist in two conformation state designed as T and R. These states are equilibrium whether or not the candy is bonded to oligomers. Means it's a form of isomers where in the form of T and R. So it is correctly binded agada, it will be in the active state. Uh, Another side get bind adaga, it will be in the inactive side. So that can be differentiated here. So this is the example for the ligand binding site and the fraction of protein in the R state. So here ligand can bind to promoter in each conformation, and the conformation changes alter affinity to promoter ligand. So here two binding sites are present here in this conformers are two sites T and R formation of protein it can be seen. So it is having four binding sites in this. So the binding of ligand can cause change in the equilibrium leading to activation or inhibition of the protein. So when the drug molecule, see for example, this one is taken as a drug molecule, when it is binded to a right binding side of the protein andaga, it will be in the active state. When it is in not binding with the right one andre, it will be in the inactive state. So this lead to the activation or inhibition of protein. So this lead to the protein activity and substrate inhibitions andre, for example, whatever the substrates are nothing but the drug molecules, ligands that can be denoted as substrates. So if the concentration of the substrate is increased, the rate reaction rate also increases until certain rate. Later on it decreases with increase in the concentration of the substrate. So substrate this is S denoted by S. So here ES2 is less active than ES. So enzyme plus substrate gives enzyme substrate further forming the product. So one more substrate when to the same one day protein ಎಂಜೈಮ್ಗೆ ಅಂದ್ರೆ ಇಲ್ಲಿ ಯಾರ್ ಇಟ್ ಈಸ್ ರೆಪ್ರೆಸೆಂಟೆಡ್ ಅಟ್ ಒನ್ ಇ ಅಲ್ಲ ಒಂದೇ ಒಂದು ಪ್ರೋಟೀನ್ ಇರೋದು ಒನ್ ಸಬ್ಸ್ಟ್ರೇಟ್ ಕೊಟ್ಟಾಗ ಇಟ್ ಗೆಟ್ಸ್ ಬೈಂಡೆಡ್ ಟು ಫಾರ್ಮ್ enzyme substrate and further forming the enzyme and the product. So one more same for the same reaction if you are going to add parallelly one more substrate then it forms ES2 means two substrate with the single protein or single enzyme that can be seen here. So if the reaction taking place in this way and daga, ES2 is less active. So ES2, this ES2 will be less active compared to this, where the second substrate cannot be binded inside the protein. Ala. So this is what the theory says here. And next coming to the effect of chromium iron zinc. So while addition of chromium to receptors or protein ಏನಾಗುತ್ತೆ when chromium is added the rate of reaction also increases, that can be seen here. So chromium, why you are going to use chromium, some of the metals here andaga we are going to chromium andaga it acts and tyrosine kinase activity. Tyrosine kinase activity andre it is nothing but signaling agent which acts inside the body. means signal production process, means in that section signaling agent inside the chromium, so inside the body. So also chromium improves glucose metabolism and resume lipid profile without diabetes. So these are the activities of chromium. And next, coming to the iron, here the oxidation of glucose by enzyme catalysis. The glucose oxidation influences the rate of reaction. Fe2 plus ions to the oxidation reaction, means when glucose oxidation andre, and the glucose react with the molecule of oxygen to produce carbon dioxide and water. So when this reaction is taking place in the addition of Fe2 plus ions, the rea- rate of reaction will decreases. So for example, in glucose oxidation ತಗೊಂಡಿದ್ದಾರೆ ಎಲ್ಲಾದ್ರಲ್ಲೂ. ಗ್ಲೂಕೋಸ್ ಆಕ್ಸಿಡೇಷನ್ಗೆ ಕ್ರೋಮಿಯಂ ಆಡ್ ಮಾಡಿದಾಗ ಏನಾಗುತ್ತೆ, ಐರನ್ ಆಡ್ ಮಾಡಿದಾಗ, ಜಿಂಕ್ ಆಡ್ ಮಾಡಿದಾಗ ಏನಾಗುತ್ತೆ, ದಟ್ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಶೋ ಇನ್ ದ ಪ್ಲಾಟ್. So the rate of reaction increases with respect to chromium, and Fe2 plus iron they increase ಮಾಡಿದಾಗ the rate of reaction decreases. And coming to the zinc oxide, zinc oxide ಅಷ್ಟೇ, the rate of reaction will increases. Same in the glucose oxidation reaction ಅಲ್ಲೇ ions add ಮಾಡಿದಾಗ glucose oxidation is nothing but the glucose react with molecule of oxygen to produce carbon dioxide and water. So that is with respect to zinc oxide. And what are the advantage of Fe2 plus the ions andre, where it can be used andre, insulin deficiency, when the insulin is very low and acts as an insulin resistance. And hepatic dysfunction, hepatic dysfunction andre, it's nothing but acute liver failure, it's nothing but the mild liver failure. So hepatic dysfunction andaga. And also coming to the zinc oxide, the as the concentration of zinc oxide is increased, the rate of reaction also will be increased. And next, with respect to effect of zeolite, when aluminum phosphate is used as a catalyst to inhibit oxidation of glucose in the presence of sodium hydroxide medium. So aluminum phosphate add ಮಾಡಿದಾಗ ಏನಾಗುತ್ತೆ, so the rate of reaction will decrease. Why ಅಂದಾಗ in the presence of sodium hydroxide in NaOH ಇದ್ದಾಗ the transfer of OH minus ions from the solution to zeolite porous decreases the concentration of OH minus in the solution. Hence the rate of reaction also decreases. And also with respect to UV light, the reaction is taking place under the UV light andre ಏನಾಗುತ್ತೆ, which can affect the reaction rate. Here also the rate of reaction will decreases. ಇಲ್ಲಿ ಹೇಳ್ಬೋದು why ಅಂತ, the by causing damage to enzymes. UV light can form cause the damage to enzyme to active site and altering its shape. So the protein though, enzymes are nothing but the proteins where it can cause damage and active sites by altering its shape. The shape of the active sites change ಆಗ್ತಿದೆ ಅಂದ್ರೆ the ligands or the drug molecules what we are taken, it won't fit inside the protein. So this can be seen here because of UV light. It can damage the proteins. So this can reduce the or inhibit the enzyme activity. So the activity of the protein will decreases, leading to decrease in the reaction rate. So as the reaction rate decrease adaga, the activity of the protein also decreases. So and one more point, UV light can also be used, can cause the formation of harmful byproducts or radicals that can further inhibit enzyme activity. So it can also formation of harmful byproduct. So for example, here you can see the plot where zinc oxide in the presence of UV light, in the absence of UV light. In the presence of UV light, the rate of reaction is increase. Actually, if the zinc oxide is not added to this, means the plot will be of change. Plot will change ಆಗುತ್ತೆ. ಏನಾಗುತ್ತೆ? In zinc oxide ಇಲ್ಲ ಅಂದ್ರೆ the rate of reaction will decreases in the presence of UV light. So when the in the presence of UV light, when the zinc oxide is added, the rate of reaction is increasing. So this is what in the presence and absence of UV light you can be seen. Addition of zinc oxide increases the rate of reaction. So here zinc oxide acts as an photocatalyst. So this is the end of the unit and summary there. You go through this whole unit to summary ಇದೆ. ಏನಾದ್ರು ಡೌಟ್ ಇದ್ರೆ ಕೇಳಿ.